Okay, so the reason it's very hard to it's very complicated to study lung cancer is first it takes about a quarter of the cancer-related death, and then normally there's not many noticeable symptoms in the early stage of the lung cancer. So even though we just take some kind of medicine or do the therapy, the prognosis of the lung cancer is not very good compared to the other disease. And the main reason I got interested in this project was when I searched for some kind of biological literature involving maybe different type of microRNA in non-small cell lung cancer, and search for some kind of mathematical model using ordinary differential equation. For this case, it was very hard to find out. So that was the main reason I got interested in. Okay, so we already just heard about the microRNA yesterday. So the main role of microRNA will be either just it'll induce the degradation of the mRNA, or it'll block the translation, and then it'll take charge of the cellular differentiation, apoptosis, or cell growth. And what we really think microRNAs are really important is because it can function as a tumor suppressor or oncogene. So depending on the cancer-specific situation, it can actually block the cancer, or it can help to, it can induce the cancer. So that's a very interesting feature. So what I want to do is actually, for example, I'm going to talk about the lung cancer today. And then there are specific microRNA. We can see that under the lung cancer situation, their expression level can be dysregulated, which means that either they can be increased or decreased. So based on those kind of their dysregulated level, I would like to suggest several microRNA as a biomarker of the cancer research over here. Okay, so here are several examples of the cancer and leukemia over here. So here is lung cancer and then liver cancer, and those are the leukemia. And then each disease has two sets of the microRNA. It's a very small font, so I'm not sure that you can see that. The red ones are the collection of the microRNA, which will be increased in the specific cancer situation over here. And then blue ones are the ones whose expression level will be decreased over here. So there's a LAS7, which I'm going to talk about today in lung cancer. So this one is already researched very well, and then it will be downregulated in the lung cancer. But if we just see the leukemia over here, then there's the same microRNA LAS7. So over here, so in the lung cancer, it is supposed to be downregulated. On the other hand, in the leukemia, it is known that it will be increased. So the expression level or just behavior of the microRNA will be really cancer-specific or the disease-specific. Okay, so there will be just two microRNA in my mathematical model. One is called MIR9, and the other one is LAS7. So the reason just we got interested in this microRNA is the biological collaborator actually do some kind of experiments. So what he did was actually he just looked at 140 patients with non-small cell lung cancer, and then compare the one type of microRNA expression, MIR9 expression, between the two cell types in each patient, one in the cancer cell, and then the other one is just in the normal cell, very close to the cancer. So, so by comparing those two cases, it'll give more verification. And then what we can see over here is that when you compare the MIR9 expression just in each patient case, then the cancer cell has about on average 30 times higher than the MIR9 expression in the normal cell. So we can see those are very high compared to the case with case in the normal cell. So those are the average data. So based on this experimental data, we can think maybe MIR9 may function very importantly in the lung cancer. And then in the later slide, I will try to include that one to suggest a very simple signaling pathway. And then we'll look at mathematical model very shortly. Okay, so here's a very simple signaling pathway starting from EGF receptor complex as an input over here. So it involved like several genes and protein like RAS, MYC gene over here. And then there are two microRNA, one miR9 and LAS7. And then both will just, either induce or block the E-cadherin. And then MMP is the final product over here. And then that will be a measure of like the level of epithelial mesenchymal transition. So, what if we look at like this pathway, it has like two negative feedback over here. And then once MYC is overexpressed, then it'll increase the miR9. And then it'll just decrease like E-cadherin. And finally, the final product level will be increased. And then if we look at the other pathway involving LAS7, then it'll block over here and increase. So both pathway will play the kind of same role to increase the final product over here. So the reason like we try to include those two specific microRNA will be miR9 came from the experimental data. And then LAS7 is already well studied in the non-small cell lung cancer literature. And then it is already known that when there's non-small cell lung cancer, this level will be downregulated. So with several hypotheses, we constructed this kind of simple pathway. Okay, so here is a mathematical model. It's a system of ordinary differential equation with maybe A species over here. And then we have like either inputs or several genes or proteins are described over here. And then M and L are miR9 and LAS7 concentration, those two microRNA. So when we constructed this model, the first three equations, we used the Michaelis-Menten kinetics because those like three species will have either inactive or active form. And then for the rest part, we use just a law of mass action kinetics or just Hill type function in case something is activated or inhibited. Yep. No, actually, that just came from when I just tried to, that kind of setting up came from based on the simulation results. Yeah. Yeah, so I haven't had a chance to check whether we needed those kind of high like Hill coefficients, based on the biological literature. Okay. So, the signaling pathway is relatively simple. And then the model just used like system of ordinary differential equation. So for me, the hardest part in this project was finding all the parameters. I have no idea like which parameters are appropriate. So, and then there are a bunch of parameters we should decide, including the initial condition, and then several rate constants, including control and saturation constant. So a lot of them are just estimated based on the simulation results. And then some of them are borrowed from the other biological literature. Maybe some concentration is known, maybe in human cell or some other type of cell. I just try to borrow to match those ranges over here. So next slides also have like a bunch of parameters, and then a lot of them are just estimated. So the thing is like a lot of like 14 parameters are actually estimated, and then maybe we were not sure that our estimation is like correct or it makes sense. So I was trying to do some kind of sensitivity analysis in the next, maybe that one comes out later. So I will talk about the sensitivity analysis in the later slide. Yep. Yeah, we want to just connect the EMT in lung cancer with microRNA expression level. Yeah, so really specific. No, just it's more towards to metastasis. Okay, so using that mathematical model, what we want to do is like try to simulate based on the two scenarios. So it is known that 10% of the non-small cell lung cancer comes from the EGF receptor, either mutation or overexpression. And the remaining 30% of that comes from the mutation of the RAS or overexpression of the RAS. So try to manipulate those two scenarios in the model. the EGFR overexpression can be simulated by actually by increasing the input variable E over here. And then to simulate the second scenario to express RAS overexpression, I try to change the catalytic production rate of the RAS, which is mu R over here. Okay, so just we already know that MIR9 should be upregulated and the LES7 should be downregulated. And in overall, the MMP, the final product, should be increased based on the biological literature, and then try to see whether our model can capture that factor. So here in the x-axis, those E naughts are the baseline, actually the baseline parameter or steady state concentration of the EGFR complex. And then we take the ratio, then this 10 means like we increase the input variable E as 10 times higher than the initial points, and then 20 times higher than the initial points. And then first figure is for the MIR9 and LES7 and MMP. So as we can guess, if we increase the if we overexpress the EGFR complex, then MIR9 will be increased, and then LES7 will be decreased, and MMP will be increased. So we can do the same thing, similar thing for the second scenario. So now, like just compare with the baseline parameter for the catalytic production rate for RAS. We try to increase that maybe 10 times higher or 20 times higher. Then we can see the similar behavior over here. So we can explain those kind of basic known feature using our model. Okay. So I showed a bunch of the, 2 tables with bunch of the parameter, and actually the 14 parameters among them are all estimated. So what we are really interested in is actually whether the MIR9 is whether the MIR9 and LES7 can be used as the biomarker based on the upregulated level of MIR9 and downregulated level of LES7. So we want to know that how those ratio between MIR9 over LES7 is correlated with different parameters. So what we tried to do was like just use the method of partial rank correlation coefficient. So try to compute the correlation between each of the parameter and the variable we are interested in. So what I was trying to do was actually there are baseline of the parameter set. So starting from that baseline of the parameter, just take the interval, maybe starting from the half of the parameter and twice times of the parameter, and then sample all the parameter just sample the all parameter in different interval at the same time. So we kind of give perturbation at the same time, and then run the simulation actually for this figure 2,000 times, and then see how they are correlated. So what we hope is actually there should not be some kind of weird correlation over here because we estimated the parameter. And if we look at this figure for mu C and mu M, they are kind of very strongly correlated. And then this can be explained based on our model because mu C and mu M were the production rate for MIC and then MIR9. So it can be, those positive correlation can be explained naturally in that sense. And if we also look at the strong negative correlation between the ratio between M and L and Km, KL, and MUL, then those are either saturation or control parameter for MIR9 and LES7. So this can be explained. And then the negative correlation between the variable and MUL, so that was the catalytic production rate for LES7. So it should decrease like this manner. Okay. By the way, just among like 14 parameter unknown parameter case, I only chose like 6 of them because they are statistically significant. Okay, so like I use like a system of ODE model and just try to just simulate the 2 scenario and just did some kind of sensitivity analysis up to the previous slides. And then now I try to extend the model. So what I'm interested in is actually there are not many things that are known about the network around involving those like each microRNA. So I will try to put some kind of random perturbation from the outside environment to each equation of the microRNA over here. So instead of using the component of ODE, I try to substitute like the microRNA part like with some kind of random perturbation over here. And then try to replace the less 7 equation by the equation with random noise over here using stochastic differential equation. So one by one and see what happens. So there are two panels over here. Top panels are the case when we put the very small noise. Like here, the sigma is like 10 to the power of -5. So when we add this amount of noise in the equation for mirror 9, so it just fluctuate very small. And then the green trajectories are the solution for the deterministic case with no noise. And the red ones are the case with noise. So since like the level of noise is very small, it'll just keep following very close to the deterministic solution. Okay. And then if we increase the noise level a little bit more, like 1.2 twice more, from here to here, then it's just stability of this concentration level already just start to break down. So those two just behave totally differently. So what we can conclude is that the mirror 9, our chosen signaling pathway is somewhat robust with respect to the mirror 9 perturbation, but not actually strongly robust over here. Yeah. So you mean the over here?
Yes.
Oh, so actually it's it's a stochastic simulation, and we just give perturbation with Brownian motion. So those perturbations can go up or down. And then if the system is not strong enough, then actually this kind of things happens. Then maybe if I try to maybe give some kind of force so that all the trajectory goes up, then I was not sure. There are some kind of discussion about those kind of like whether we should keep maintaining the positivity when we use like Brownian motion, but I was not sure about that part. So I just said, for now, I just use this one directly. That's a very good question because I've never thought about that. Okay. So if I try to add the noise to the equation or try to multiply.
Well, it would get around figure detection, I think, because if you use probabilistic parameters, the positivity
No, so you mean like just using some kind of exponential form of the Brownian motion, something like that, or? Parameter. Then the question is like which parameter? Because what I was trying to do was like actually the perturb like a mirror 9 in some sense, perturb less 7 in some sense. So.
But the mirror 9 is part of the network, right? So because you want to change the characteristics of the network that's the basic mirror 9 rather than some outside perturbation. This is like adding some mirror 9 to the system.
Well, I think, we don't really know how mirror 9 connects with other things. We don't really know. So the reviewer wanted to see wanted to see to what extent this is stable. How will some noise affect the results? Yeah. So we just take, we don't really know. So we just take, to illustrate, just the white noise, which is the most standard one.
Maybe I thought that would be okay because we are more interested in some kind of noise from the outside.
Yeah,
so that's what I did, like over here.
Because we really don't know. It's not known biologically how to connect.
Okay. Yep.
And what is it the deterministic values on the y-axis for the MRI, or the number is very small?
Yeah, number is small. So so unit actually, if we go back to the table of the parameter, there should be somewhere the steady state level of the MMP in the previous slide. So actually, just when I search for the steady state concentration for MMP, actually this case is for mRNA, and then I can only find one reference with a very small number. So we just change the parameter in that way. That's too small.
Yeah, because
That's
it's
in the order of a few molecules per cell. So you want to fully subtract it.
Could it be? Yeah, I thought of maybe just we can try to do. Try to use stochastic model in that case, but yeah, but as a starting point, we can look at the deterministic case first, right? Yeah, actually, there are several things like I just wanted to suggest to fix, so that will come out in the discussion or future work. So you can give me more information, more opinion about that. It's very.
if you have cells on one side and you have cytokine on the other side, the parameter will be very negligible. But if you non-dimensionalize, then it will become a regular O1 number.
Okay, but not for all of them. That's what
But on the parameters which you estimated, for something estimated, you have all these precise values.
No, that's right. That's because the reason is because the estimate is based on some assumptions.
Steady state, actually based on the steady state concentration. So under the assumption that maybe without this kind of like species, so maybe we can actually just plug in the steady state concentration and then try to compute those kind of parameters. So some of them are derived from there, and then some of them are just came from during the time of when I just simulated, try to choose the better parameter.
But
So
for
the
some,
estimation comes out of some assumption, and you compute based on this assumption, you get this number.
so it's not a complete guesswork.
No.
It's not an estimate. It's just estimated.
Okay, so where did I stop over here? Okay, so I talked until the moment that we try to extend the model by replacing the ODE part by using stochastic differential equation over here. So when I just replace the equation for the MIR9, then that one I already explained. And then let's move on to the second case. So if we perturb the less 7 concentration, then it shows a little bit different, because when I perturb with a small noise, then it fluctuates around the deterministic trajectory. And if I increase actually twice times higher noise, then actually the fluctuations becomes large. But when I simulate this one many times, more than several thousand, and make the average, then still the average actually keep following the deterministic trajectory very closely. So what we can conclude is that in the previous slide, this network was somewhat robust with respect to the MIR9 perturbation, but not strongly robust. In this case, we can conclude that our network is very strongly robust with respect to the less 7 perturbation over here. Okay, and then I was trying to find out the reason from the topology of the signaling network, because if we look at the network around the MIR9, then noise over here, upstream noise can directly just go downstream level over here up to the MMP. On the other hand, if you look at the network around the less 7, this one involves 2 negative feedback from here to ROS and then arc to SOS. So that may be the reason, so that the upstream level of the fluctuation can be damped out, and then the system becomes more robust with respect to less 7 perturbation over here. And then the biological collaborator actually tried to confirm this model, and then see whether the experimental data can give the consistent result with the mathematical simulation. But there was some kind of problem, because gafitinib is the inhibitor of the EGF receptor. So if we increase this dose, that means we decrease the input. So as we go from the left to the right along the x-axis, actually the miR9 is supposed to be decreased, LES7 is supposed to be increased, and then overall MIG expression should be decreased. But if we see the data over here, then miR9 downregulation can maybe in some sense captured over here, even though it's not monotonic over here. But if we see the LES7 expression, then it is supposed to be increased based on the model, but it does not increase at all. So what we can say is maybe we need to find another missing component of the network involving LES7 so that model can better represent the data over here. And then MIG expression, it is just decrease over here. And then this one maybe does not move very much, but those two can be, maybe we can say that it's a decrease. So still there is a lot of gap between the model and the data. So far what I tried to do was like just briefly introduce lung cancer and microRNA and try to suggest a very simple network involving two microRNA, miR9 and LES7, and try to construct the mathematical model using ODE over here, and then run the two simple scenario in the case of EGF receptor overexpression and RAS overexpression. And then I tried to extend the model by replacing the ODE part using stochastic differential equation to see whether the network is robust or not with respect to miR9 or LES7 random perturbation. The thing is, actually, we need to still find out the missing component around the LES7 to match the model with the data. And another thing is, the overall reason of this mathematical modeling for me was try to use like those two microRNA as a biomarker, which means that if we can find out the just upregulation of the miR9 and downregulation of the LES7 at the same time, then probably we can say, probably this will tell us that this patient has lung cancer. So that's going to be the final goal. But now, just the whole network is somewhat robust with respect to the miR9 perturbation. So the final goal will be find out the better modify the model so that both the whole network is strongly robust with respect to the random perturbation of both microRNA. That's going to be the final goal. And then this type of approach, like using the signaling pathway with like microRNA, can be applied to different type of the cancer or other cancer-related disease. So that could be another approach for future work. So this was a collaboration with Evne Friedman and Patrick Nanasinkam. And then thank you very much.